So our third plenary speaker is Guoping Feng. Guoping Feng is the Poitras Professor of Neuroscience in Brain and Cognitive Sciences and in the McGovern Institute. And Guoping's appointment and recruitment I regard as a coup for MIT and for BCS. Guoping started his lab here last June, and we have been thrilled by already the impact that Guoping has had on our community. So, Guoping.
Thank you, McGovern. I'm very happy to be here to give you an overview of the current status of psychiatric research. So you probably all know that mental illness, psychiatric disorders are a very big problem. Right? So they are estimated about 400, over 500 million of people around the world has psychiatric mental illnesses, and nearly 1 million people every year commit suicide. And they are, this is one of the leading, as a group, this is one of the leading causes of disability lived with life. And this is probably most surprising, most astonishing fact is 1 in 4 family has at least one member has mental illness, and so they affect not only the patient but also the family members, because most of the care are, in many of the cases, are cared by family members. So there's a huge burden on the society. So as a group, neuropsychiatric disorders are the largest basically cause burdens of disease worldwide for disability, lost days of days basically lived with a disability. So this is a huge problem. Despite this huge problem, and have been recognized for decades, but there's not many options of treatment. So this is the slides Ed Skolnick, our director of psychiatric research, gave to me recently, and I used actually a few of his slides of this data. So basically, in the past, 60 years, there's a significant, dramatic increase in the, for example, drugs that with no mechanism of treating heart disease, but there's almost no change for drugs for treating, depression and schizophrenia. So the problem is as big or even bigger than before, but the treatment options are very few and very effective. So why? It's not like pharmaceutical companies are not interested in developing. These are considered commercially a very profitable, targets. The problem is that one of the biggest reasons, the main reason actually failed, or billions of dollars have been spent by pharmaceutical company and the research institute, but failed to develop a drug. The main reason is that we actually don't know the mechanism, what causes disorders, and what are the underlying pathology of these disorders. So there are many examples. You can say, pharmaceutical companies spend billions of dollars to try to develop all kinds of drugs, but and most of them are fail. The reason is, it's just a guess. As a guess, then there's no understanding of disease. There's no good animal models. So what do? You in the lab start a test tube, or at the most you culture a cell, then say, okay, what do you do? Effective, let's start clinical trials. And so it's basically a black box. Let's see, spend $20 to $200 million to see whether clinical trial have effect or not. So most of them you can get, although we are all very smart, and pharmaceutical company scientists and the CEOs are super smart, but it's just a guess, right? So most of them fail. Just as Li Hui showed you, most of our clinical trials for Alzheimer's disease have failed. So this tells you the most important thing for develop treatment for any of the disorders, especially for brain disorders, is to understand the underlying pathology of and underlying neurobiology and neuropathology of these disorders. So how are we going to figure out this underlying pathology and the cause? So for psychiatric disorders, there are very strong evidence that there are genetic factors that contribute to these disorders. This is a chart just show you that in general population, for this is just for bipolar and schizophrenia, it's around 1 to 2% of incidence. But in the family members, between parents, siblings of children, of any of the family, if it has disorder, then the chance of their siblings or parents or children are much, much higher incidence. So it means there's a very significant contribution from genetic factors. So previously, 10 years ago, 10 years ago, it's almost impossible to think about how we're going to figure out all these differences, because each genome, everyone sitting here, there are many, many differences. Right? There are, thousands or millions of differences. How do we going to figure out? So, but now there are ways you can do, right? It's because of the advance of technology. So, basically, based on that, the key thing to the future of psychiatric research in developing the last, probably mostly in the last less than 10 years, 7 or 8 years, is on the two fronts, which is genetics and genomics, to figure out the genetic contributors or causes or risk factors that can lead to psychiatric disorders, then use that information to help us to understand if you have that risk factor or have that mutation, what happens to the neurobiology or neural circuits and pathways in the patient brain that we can potentially correct. So the frontiers basically we're talking about is genetics, genomics, and neural circuits and pathways. And so for the genetics, basically the new genomic technologies are driving the genetics. So now we can, the technology started with whole genome, human genome sequencing. Right? And the prediction at that time when it started would take 20 years to sequence one human genome, but now you can do it in probably weeks to solve that. So this dramatic advance of technology, which we have a huge advantage here at MIT in collaboration with the Broad Institute, is that, and that give us the most current up-to-date knowledge on the genetic causes and genetic factors involved in psychiatric disorders. So we basically now with the technology, if there's enough funding, and you can basically identify all kind of variations between humans and specific patients and a specific group of disorders. So that gives us a very strong and foundation to start to understand what is going on with the neurobiology of these disorders. So with the genetic basis, then it will give us a tool, basically can use animal models or model system to say, if we have a mutation in this patient we identified that can contribute to this schizophrenia, obsessive compulsive disorder, autism, or bipolar disorder, and we can use a model system to try to figure out what is what went wrong with in this brain. And that the hope is that by understanding the neurocircuitry defect and the pathways, then it will help us to understand figure out a way to treat them to correct this problem. So I'm going to use an example in my lab, which we have been using mice as a model. There are many other model systems can be used to study, and we will have a panel to discuss this about these different models. And we'll use a simple example in my lab to connect from genetics, human genetics, to animal models, from behavior to understand neural circuits, to illustrate how we can go on to understand these most debilitating and complex disorders in the brain. So one of the main areas in the last few years have emerged is that many of the defects are concentrated in one places. Basically, is the place where the neurons and neurons communicate. Because if we think about whatever we do, whatever our behavior, our emotion, all depend on neural circuitry activity. And we have billions of neurons in the brain, and they don't randomly connect. They have very precise connections. And then they also have very precise, at a millisecond scale, very precise communications, right? If you do one particular thing, there are a group of neurons activated, and precisely this neuron sending signal to activate different specific muscles, so you can raise your left hand or right hand. And you can think about all other, your speech, your hearing, your behavior, your anger, your emotion. Everything is controlled, basically related to neural circuitry activity. So these all depends on neurons communicate, hundreds of, thousands of, millions of them. So most of the neurons communicate through a very small apparatus, only a couple micron size, called synapses. So because the neuron-neuron communication is critical to our brain function, you can imagine this is basically a hotspot for disorders. And So neurons, these are just drawings of how neurons make connections. So this is basically the receiving neuron. There are many other neurons send their axons to make connections, and these are the site called synapses. They send signals, chemical signals through this vesicles, then release, and then the receptor on the other side will receive a signal, turning into different electrical signals and the chemical signals, leading to gene. Not only the response, electrical response, but also gene transcription changes that you heard from Li Hui talked about in the Alzheimer's disease brain. So in this little teeny tiny space, this is a drawing of one of the synapse, which is at the postsynaptic side, just this little receiving side. Actually, this is about a couple of microns at the very tip, which is about 100 to 200 nanometer size. There are up to 1,000 types of proteins clustered there to make sure our brain functions properly. So it says two things, right? One is that the brain really, this synapse, although very small, does a lot of things, right? There are many different ways to modulate it, regulate the function. On the other hand, if you have so many things needed to regulate function, you also is a trouble spot. Right? Many of these genes mutation can cause a problem for the synapse, and they may cause a lot of problems for psychiatric disorders and the neurological disorders. So in my lab, like many other labs, we have many labs around the world are very interested in how these synapses are assembled, how their function are regulated, and what do we go wrong if some genes are mutated, and what do we go wrong with the synaptic function, and what do we go wrong with their behavior in the whole animal. So we are focusing, I'll give you one example, and then I'll give you an example on this protein. So this complex we call a scaffolding complex. They consist of several groups of proteins that are linking to each other, that they form a scaffold. So now when you have scaffold, then you can bring other proteins into the synapse and assemble a large macromolecular structure that will help to function at the synapse and also modulate the synaptic function, because our synapse, our brain, is not only it's not a function properly, but also it's very dynamic. Right? It's not like a static. Once we make a wire connection in the computer, it never changes. This is change. It's constantly changing, constantly dynamic. We can learn things, we can eliminate things, we forget things. All these are very dynamic processes. All these processes, most of it happens at the synapse. So this complex is very important for assembly of the synapse, but also bring all the signaling proteins into this synapse to modulate synaptic function, which means that if we figure out what's going wrong here, we can also find a way to modulate this synapse. So maybe we can find ways to correct the problem in psychiatric disorders. So one of the protein I will show you how disrupt this protein called SAPAP, one of the family members can lead to abnormal behavior, and how we can correct this problem. So there are 4 families, right? And one of them called SAPAP3 is highly expressed in one area called striatum. You heard that from Miriam's talk about striatum, which is control almost all movements and also behavior, and this is the brain area that control the movement and a variety of behaviors, especially related to repetitive compulsive behaviors and in obsessive compulsive disorder, in autism, and also in Tourette's syndrome and trichotillomania, all these with repetitive compulsive behaviors in psychiatric disorders. So when we mutated this in mice, what you can see is that these mice have repetitive compulsive behavior. This is a mutant mouse. We took it out, put on the cage top. And usually, some of maybe have experience, you take a mouse out of a cage, the first instinct they go is run away from you, right? Your instinct is to try to catch it. But this mouse is a mutant mouse. You put it on the cage top, it can just stay there. It stays there, and it seems the grooming, compulsively grooming itself, is more important than the basic instinct to run away. Right? So you can actually videotape it. So this type of, next slide I will show you is very, graphic, dramatic. So basically, this constant grooming and lead to a gross bloody facial lesion. So they're basically grooming their hair and skin off. Then remind us that obsessive compulsive disorder. So some patients, they will wash their hand 500 times a day. They're bleeding. They still cannot continue. They still want to wash their hands. So this is so when you videotape and to look at what do they do during a 24-hour period. Right? The normal mice they spend less than 10% of total their daily time grooming. The mice are like humans. They clean their skins, they clean themselves. So we also clean. We probably don't spend 10% of our daily time to clean ourselves. So mice are better in that way. But on the other hand, right, so these mutant mice, they basically spend a third of the day grooming themselves. So they cannot shake off this grooming, compulsive, repetitive grooming. So that leads us to think this may be a model that, based on the expression in the striatum, which has been used in clinical studies in OCD patients, has been strongly implicated in OCD, in obsessive compulsive disorder, and in autism. So we decided to look at whether these mice also show the symptoms or phenotypes similar to other aspects of OCD. Right? OCD is an anxiety spectrum disorder. Most of patients have a dramatic increase in anxiety. And we use in mice, there are very standard ways to test their anxiety-like behavior, and we show that these mice also have dramatic increase in anxiety, which is very similar to what OCD patient experience. And then there are no specific treatment for OCD. There are actually no specific treatment for any major psychiatric disorders because we still don't know what causes them or what's the underlying pathology. But the most frequently used drug, which are partially effective, is Prozac type of drugs, actually. And so we tested these mice, and we can see that these mice are very effective in patients. These are only partially effective in about 50% patients. So the other 50% basically they have no treatment, and the 50% which are about 50% which are effective, only partially effective, usually there's no cure for them. But these mice also, when we treat them with fluoxetine, Prozac, they are also partially effective in control some of the symptoms in these mice. So this kind of test led us to believe that maybe these mice can be used as a model to figure out what is going wrong, what is going on in the brain of these mouse actually lead them to groom themselves, that groom their hair and skin off. They'll still keep doing this. A symptom which is resemblance of OCD-like behavior. So one of the key things is that there are many studies have shown suggests that the communication between our cortex to the striatum, then to the thalamus and back to the cortex, this loop, neural circuits, is involved. But there's no direct evidence that defect here actually can cause OCD. Because you cannot just go doing any of these lesions or damages in patients to test this. But in animal models, you can actually, not only you can test this, but you can also try to, can we fix this by re-expression these genes into this area. So the first thing we did is to use electrophysiological recording to show that indeed there's a dramatic defect in communication between these cortical cells to the neurons with the striatal neurons. And then we can say, this is mutant mice. They are lacking of protein. They have synaptic defect, neuron-neuron communication defect in the striatum. Can we prove that the defect in this area called striatum is the key? And so the way we can do is in mice is you can selectively express this gene only in this area to correct the problem. Not everywhere else still has a mutation, but only in these neurons you express gene. Can you correct the problem? The answer is that yes. So when we put this control virus into this area, and these mice still have lesions, they're grooming still like, I call them crazy mice. And so they're still grooming them, like grooming their hair and skin off. But in the mice, the mutant mice, if you put the gene back only in the striatum, in that brain area, and it does not do that anymore. So these mice actually, you basically cured this problem. So they give the direct evidence that a defect in this brain area, striatum, that the synaptic defect, which means the neuron-neuron communication problem defect, is the causative to this problem. Now you have a direct evidence that is the problem. Now we get us excited. Maybe we can find a way to treat, better way to treat a patient. The problem is mice are not humans, right? Maybe there are many studies in mice may be effective, but they are not may not be effective in humans. So how do we know this gene, this type of mutation actually contributed to human patients? So now with the genetics that I mentioned earlier as a key aspect to advance our understanding of psychiatric disorders is we cannot go back and use the sequence technology to sequence this gene in OCD patients. We actually did that, and we found we found quite a few we call the variants, basically genetic differences between the OCD patients and the normal people. So these things, we call the variants, genetic material differences between the two groups. And so with the mice, give us the opportunity. But what we don't know is whether these are causative to the patient, right? You cannot go recreate this in humans, but you can go recreate this in mice. So what we did is we can create this exact mutation we found in human, and create this mutation in a mouse gene, because the mouse gene and the human gene in this gene, in most of other genes, are almost identical at the protein levels. So we show that this mutation we found in humans, OCD, actually complete loss of function. So which suggests that a single nucleotide, one nucleotide change, right, that lead to one amino acid change, actually completely lost, basically lead to the complete loss of function of this protein. So now we actually made a mouse. Instead of mutated the whole gene, just put this mutation, a single mutation found in OCD patient, into a mouse. And we're waiting for the mouse. If the mouse actually shows a very similar phenotype, like OCD-like phenotype, then it tells us that this mutation we found in human patient probably will contribute to the OCD in that patient. This is probably the strongest one of the strongest evidence you can ever get to prove that a genetic variation you found in a human patient actually may contribute to the problems in the patient. The most exciting is that if we think that a circuitry defect is the key, then there are many implications. First of all, the two most important implications are, first, if the synaptic defect and neuron-neuron communication in this area is key, then we can predict many of the genes, as I said, there are about 1,000 type of proteins involved in assembly, function, and plasticity of the synapse. Then many other genes which are important for the development or function of the synapse in the striatum, they may be also very important, and their mutation may also contribute, cause the same problem. So that leads us to have a collaboration with actually Broad Institute and the Stanley Center for Psychiatric Disorder at the Broad Institute to sequence of 600 genes which are all highly expressed in this area, and sequence the OCD patient. Can we find a cluster of these mutations in this gene compared to other genes which are not expressed here? So that gives us a very strong, if it's true, it will give us a very strong evidence that genes involved in this neural circuit is critical to the pathology of repetitive compulsive behavior. Then the second prediction is that if this is the case, then you may be able to find treatment by just modulate the circuitry function. It doesn't matter which gene, because right now, if there are 300 genes involved in OCD, there's no way we can go individually treat them correctly, because gene therapy is still years away and is not trivial right now. But if it's a circuitry defect, if they cause a similar neural circuitry defect, then the only thing needed to do, use the technology Marion has mentioned, that she can figure out what are the differences between the gene expression and signaling pathways between the patient and this mouse model and the normal mouse. Then you will have a pathway. Actually, she already did this work actually in this pathway and published several papers, very nice papers. And you basically can use the current knowledge. Once you figure out the circuitry defect, use the current knowledge, actually go on to directly modulate the function activity of the neural circuitry and use a small chemical to correct this problem. So this gives us a hope that by combining genetics and neural circuitry studies, use animal model as the stepstone, maybe soon we will have, I hope soon, and this soon is not a days or months, at least a few years away, that it will have a much better approach or a strategy to treat this terrible psychiatric disorder. This is just an example of OCD. A similar experiment has been going on in my lab for autism and bipolar disorders. All these we hope to combine with many other advances in the field that eventually will give us a hope that we will have a much specific and much more effective treatment for psychiatric disorders. And so that's this is any slide will show. Maybe we'll have a circuitry-specific treatment, basically, for these psychiatric disorders. Thank you.
We have time for a couple of questions before we break out.
So what's the difference between knocking out one of the genes and just changing one of the mutations? Is that the same thing?
Technically, similar, but conceptual it's a difference. So a knockout means we completely eliminated the gene function. And in most of the mutations or variants we find in humans, they are deletions like this, copy number variations or deletions, which are not as common as the point mutation single. So the knock-ins means you can knock in any kind of mutation you find in human into the locus of mouse gene that mimic the mutation you find in humans.
So in a knockout, you're basically taking out the whole gene, but in the mutation, you're just changing a step.
Yeah, you just change a single. If it's a single amino acid nucleotide change, we just replace that nucleotide, single nucleotide, anywhere in the genome. Basically, in a mouse, you can do that now.